Muscles are the organs of the muscular system that are capable of contracting. This allows us to move. As we start looking at muscle tissue, we're going to change the name of everything you know. A muscle fiber is now what we're going to call a muscle cell. The muscle cell or the muscle fiber is filled with myofilaments. These are the proteins actin and myosin. When we're talking about muscles, you're going to see certain words crop up repeatedly. Myo, mice, and sarco are all words that are prefixes for muscle. There are three types of muscle tissue: skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscles are the organs that are attached to bones and skin. They're elongated cells that are called muscle fibers that are cylindrical and multinucleate. When you look at these cells under the microscope, they appear to have little striations or stripes, so this is referred to as striated muscle tissue. Also, this type of tissue is voluntary. We consciously control when these muscles contract. They contract very rapidly, but they do tire easily. They are very powerful in their contraction. The nervous system is required to stimulate these muscles to contract. Cardiac muscle is found only in the wall of the heart. It makes up the bulk of the heart. This tissue is also striated. It has the same kind of stripes that we see in skeletal muscle, but it is involuntary tissue. We do not consciously control when the cardiac muscle contracts. There are cells in cardiac muscle that can act as pacemaker cells. We say they are autorhythmic. They can contract without nervous system stimulation. However, these cells can be stimulated by the nervous system. The cells here are branched and they're held together by intercalated discs. We'll talk more about cardiac muscle when we do the heart. And the third type of muscle tissue is smooth muscle. This is found in the walls of hollow organs like the stomach, the urinary bladder, the airways, the blood vessels. It is not striated. It has a different arrangement of myosin and actin, but it is also involuntary. We do not control when this muscle contracts. Like the cardiac muscle, there are autorhythmic cells in the smooth muscle tissue. So this tissue can contract without being stimulated by the nervous system, but the nervous system can stimulate it to contract. So as we look at the three types, skeletal muscle has the striations. The cells are long and cylindrical, and they have multinuclei. The cardiac muscle, you see the same striations, but the cells are branched, and these darker striations are the intercalated discs. And here we have smooth muscle. The cells are spindle-shaped. They dovetail on each other. Smooth muscle tends to assemble in sheets. Muscles have four basic characteristics. First of all, they're excitable. They're able to receive and respond to stimuli from the nervous system. Second, they're contractile. They can get shorter. Third, they're extensible. We can stretch them, but the really neat thing about them is they're elastic. We can shorten them, we can stretch them, but they're able to recoil and return to their resting length. Muscles have several functions. One is movement. Locomotion is by the skeletal muscle. Blood is pumped through the body as a result of cardiac muscle contraction, and food moves through the digestive tract as a result of smooth muscle contraction. The skeletal muscles are involved in maintaining posture and in stabilizing and strengthening joints. All muscle contraction will generate heat. Skeletal muscle is going to be the primary focus of our discussion in this chapter. Skeletal muscles are organs. All four major tissue types are found in a skeletal muscle. As we look at the gross anatomy of a muscle, there are nerves that stimulate the muscle to contract. There is at least one nerve that goes to each muscle. Each nerve is made up of several neurons, and the neurons branch and go to individual muscle fibers or groups of fibers. Also, there is a rich blood supply to skeletal muscle. One artery feeds each muscle, and multiple veins drain the blood from the muscle. These veins branch repeatedly, as do the arteries, so that the blood goes to all parts of the muscle. The arteries deliver oxygen, the veins remove waste products. This is a highly metabolic tissue with a high demand for oxygen and a high production of waste products. Skeletal muscle has a number of connective tissue coverings to give it strength. This connective tissue helps support the cells and reinforce the muscle as a whole. The endomysium is the connective tissue that covers each individual muscle fiber or cell. This is mostly reticular fibers. The perimysium surrounds the fascicle. Muscles are arranged in groups of muscle cells known as fascicles, and each fascicle is surrounded by its own connective tissue sheet known as the perimysium. The epimysium surrounds the entire muscle, all of the bundles of fascicles that make up the muscle. This is a dense, irregular connective tissue. And then we have the deep fascia. This is the connective tissue that will bind muscle groups together. So there are 4 muscles that make up the quadriceps group. These muscles are all bound together by deep fascia. So as we look at our muscle, here we see the epimysium around the entire muscle. Here you see the bundles of muscle cells. These would be the fascicles, and they're surrounded by the perimysium. And within each fascicle, we have a number of muscle cells, or muscle fibers, and they're each surrounded by their own endomysium. Skeletal muscle has to be attached to the bone. They have to attach in at least 2 places. One point of attachment is known as the insertion. This is the attachment to the bone that will move when the muscle contracts. The other attachment point is the origin. This is the bone that will not move when the muscle contracts. There are 2 different types of attachments. In direct attachments, the epimysium fuses directly to the periosteum of a bone or to the perichondrium of a cartilage. Here it's like the muscle and the bone become one. Most of the attachments are indirect attachment. The connective tissue wrappings extend beyond the muscle, so all of those connective tissue fibers come off of the epimysium, the perimysium, and the endomysium and come out beyond the muscle. If this is a rope-like extension attaching the muscle to the bone, this is known as a tendon. In flat muscles, this extension will come off in a sheet-like extension, and that's known as an aponeurosis. Here we can see a tendon. These will be connective tissue fibers from all of these coverings of the muscle. When we look at the muscle fiber microscopically, each individual muscle cell or muscle fiber is a long cylindrical cell. It has multiple nuclei. The bigger the cell, the more nuclei you have to keep it under control. The sarcolemma is the name we're going to use for the cell membrane. See sarco, that muscle thing? This is the membrane of the muscle cell. The cytoplasm is now going to be called the sarcoplasm, and we're going to find a lot of glycogen stored in muscle tissue and a protein called myoglobin. Myoglobin is very similar to hemoglobin. Hemoglobin carries oxygen in your blood. Myoglobin will store oxygen in your muscle cells. There are the same kinds of organelles in muscle cells that we find in all other cells, but we do have the sarcoplasmic reticulum. This is the new name for the endoplasmic reticulum. We're going to see organelles known as myofibrils, and we're going to see extensions of the sarcolemma into the muscle, and these are going to be known as T-tubules. The myofibrils are the organelles that contain the contractile element, and they make up about 80% of the cell, so these are packed in the cell very tightly. Each myofibril is composed of many contractile units known as sarcomeres. The sarcomere is the smallest contractile unit of the muscle cell. The skeletal muscle cells appear to have striations. This is because of the overlapping of the myofilament. The myofilaments are the cytoskeletal elements that are packed into the myofibrils. The striations will have the A bands, which are the darker colored areas, and the I bands, which are the lighter colored areas. So as we look at a muscle cell, here's the sarcolemma, and here are all those myofibrils. And within each myofibril are packed all of the cytoskeletal elements. And the way these are arranged is what's responsible for the striations. So you have the dark A bands and the light I bands. You can also see that there are several nuclei in this cell, indicating that this is a multinucleated cell. The sarcomere is the smallest contractile unit of a muscle fiber. It's what we call the functional unit of the muscle fiber. It runs from Z disc to Z disc, and it's composed of overlapping thin and thick myofilaments. These are the contractile proteins. Myosin is the protein that makes up the thick filaments, and actin is the protein that makes up the thin filaments. So as we go back and look at a sarcomere more closely, here is a Z disc, and here is a Z disc. The blue lines indicate the thin actin filaments. The red lines, the thick myosin filaments, and you can see how the myosin overlaps the actin in spots, and then there are places where we do not have myosin. So here's those light I bands and those dark A bands. The actin is anchored to the Z discs. The myosin is held in place by this M line. So this stabilizes the myosin. these filaments are going to slide over each other to shorten the sarcomere. Now here is a picture that shows the sarcomere a little differently. Here's that M line that's holding the myosin filaments. And the myosin filaments, you can see, have all these heads on them. And then here are the thin filaments, the actin filaments. Your Z disc, and here is the entire sarcomere. The thick filaments are made of the protein myosin. Now each thick filament is several myosin molecules held together. A myosin molecule is a rod-like molecule that has a tail that has two globular heads on it. The myosin head is the binding site for actin on the thin filament. When myosin binds to actin, we're going to form cross bridges. Also, the myosin heads have binding sites for ATP. There is an enzyme called ATPase that helps form ATP on the myosin head. So as we look at the thick filament, here is that rod-shaped protein with the two heads. Here is where we're going to have the actin bind, and here is where ATP can bind. Now that whole thick filament is several of these molecules bound together, sort of with the tails together and the heads facing outwards, so that on the thick filament you have heads on one end and heads on the other end, and no heads right in the middle. The actin makes up the thin filament. Now this is a twisted double strand of a fibrous protein. On the actin we have the sites for the myosin head to attach. This happens during contraction of the muscle. There are two other proteins associated with actin. One is tropomyosin. This is a regulatory protein. It is in position and blocks the myosin binding sites for the myosin head, so the myosin cannot attach to the actin when tropomyosin is in place. The other regulatory protein is troponin. Troponin is what regulates the position of the tropomyosin. So here we see our actin filament, that double strand. They look like beads, and they have the myosin binding sites on each bead. And then we have the thread-like tropomyosin, and when it's in place, it covers all of those openings. The troponin is attached to the tropomyosin. If the troponin changes its shape, it will reposition the tropomyosin so that these binding sites will be available. So looking at the whole picture, here you can see the myosin with heads, the tails all pointed toward the middle, and they're stabilized by the M line. And then we have our actin fibers, and notice how the actin overlaps only where there's myosin heads, so that those myosin binding sites and those myosin heads are in position. The only reason they don't attach and make cross bridges is because of the tropomyosin that's blocking those myosin binding sites. There are a couple of more myofilaments that we need to talk about. One is the elastic filament. This is composed of a protein known as titin. This is what holds the thick filaments in place. It helps the muscle recoil after it's been stretched, and it also resists excessive stretching or overstretching. Dystrophin is a protein that links the thin filaments to the proteins of the sarcolemma. This allows the thin filaments to be somewhat stabilized so that when we slide, we'll pull the Z discs together. So here we see the sarcomere again. Here are the elastic filaments. So when the muscle is stretched, these will get longer. When the muscle has been contracted, these will pull it back into its resting position. And then dystrophin helps connect the actin to the various parts of the sarcolemma. The sarcoplasmic reticulum, what we used to call the endoplasmic reticulum, is a network of smooth endoplasmic reticulum that surrounds each myofibril. Most of these run longitudinally in between the myofibrils. There are pairs of terminal cisternae, that is, there are places where this kind of pools out, almost making like foot-like projections that will stick to each other, and these will form perpendicular cross channels in the sarcoplasmic reticulum. The sarcoplasmic reticulum helps regulate intracellular calcium levels. Most of the cellular calcium is stored in the sarcoplasmic reticulum and released when necessary to allow for muscle contraction to occur. So as we look at the sarcoplasmic reticulum, here we see it running around each myofibril, and you can see periodically it makes these channels. It kind of pools together and it makes these channels. The T-tubules, or the transverse tubules, are a tube that's formed by the sarcolemma protruding into the cell interior, very deep into cell interior. This helps increase the muscle fiber's surface area, and it allows the electrical nerve transmissions to reach deep into the interior of each muscle fiber. These are going to run between those terminal cisternae. This will allow the sarcolemma to be in very close contact with those terminal cisternae, those projections of the sarcoplasmic reticulum. The triad are the two terminal cisternae that are divided by a T-tubule. So if we look again, here we see the sarcoplasmic reticulum. Here we see the T-tubules. This is the plasma membrane, the sarcolemma, coming down deep into the cell, coming back out and staying continuous, so we don't have any breaks. And here we see the sarcoplasmic reticulum nestled very closely to the T-tubule. This would be a triad, these two cisternae and the T-tubule.